Hi U10, Mr. Seymour here. Sorry I'm not there today, but hey look, via YouTube I can still run through this summary, this revision topic with you on Fridays. So we're up to the topic 3 of our Year 10 revision lessons, and it's reviewing today Generation Martian Unit, which is all about reproduction and genetics. Now, you remember back that the first few lessons we were talking about, you were a team that had to provide and work out a team of 10 individuals, 5 females, 5 males, to go to Mars, and it was a one-way journey. So it was really difficult to figure out who those people were, diversity in genders, diversity obviously in cultures, diversity, similarities in language. So you came up with criteria by which you can filter out and find the best 10 astronauts to send on a one-way trip to cultivate and have the first Martian babies on Mars. So we're going to quickly go through not the context of that storyline, but the theory that underpinned it. So obviously we went through all this, but wrapped all that in a story about these Martians, about these astronauts. We're not going to really do that today. We'll just go through the very simple, bare basic information that you need to know that we unpacked during that one. So let's get into it. So the first idea that we went through was we need to know about DNA. So the 37 trillion cells in your body, each living cell has this strand of DNA a couple of meters long, and this is what it looks like spinning around here. So we have 2 side strands, and then you've got this middle rung, these middle rungs. It looks like a ladder, really, that's been twisted. So it is responsible for the whatever is necessary to repair and grow your body. That is what DNA is for. It is a code that allows the blueprint to be able to make things like proteins and all the other things necessary for your body to continue to live beyond your mother's womb. So that includes the bases, which is A, T, G, and C. That also includes the phosphate and sugar side arms. Next one, we looked at different types of reproductive strategies. So we said, well, there's 2 types, really, generalizing. We have sexual reproduction and asexual reproduction. Asexual reproduction, and many plants have this reproductive process, and also some very simple animals do as well, where they only need one parent, and you can copy yourself and then make offspring, as many as you like, through asexual reproduction, like budding, like cloning. Though most complex multicellular organisms and animals use sexual reproduction. So it requires, through sexual reproduction, the formation of sex cells, gametes, ovum, sperm. We also therefore need the ovum, the egg cell, and the sperm to get together, to be fertilized, so that we therefore have a simulated combining of that called embryogenesis to make that new zygote, make that new baby, the embryo. So here we have the reasons why. In humans, male and female, in order for those gametes that I talked about before to meet, and we talk about internal fertilization as well, rather than external fertilization, using the gametes via the testes in the males, and in the females we have a pair of ovaries, 2 testes and 2 ovaries. Then we went and looked at the structural components necessary for these gametes to be formed and also collected in the correct area where they can come together. So in order to do that, those structures are as follows, and we went through the reasons why the male and female reproductive organs need that. And it's interesting, just think, if you remove any one of these structures, so penis, urethra, scrotum, testes, seminal vesicles, and vas deferens, if you remove any one of these, then the likelihood of fertilization of that egg from the sperm in the mother's womb, in the uterus, is really depleted. So it requires the entire structural components of the male reproductive system for that to occur. Also, we looked at the parts of the sperm which are really necessary, because if you have only what you see on the screen here, and then the sperm is just a single circle, a little cell without the tail, then it wouldn't be able to move through the upper levels of the vagina and through the uterus to get to and up the fallopian tube to get to the egg for fertilization. So it really needs the sperm really needs that tail as well. And remember, in between the head and the tail is this little amazing small little machine that spins that tail of the sperm around. It's got a lot of mitochondria in there. That's where the energy comes from, for that tail to just move around, for that sperm to have motion and go forward. So we won't spend too much time on this, but just as a reminder, I will upload this PowerPoint. These are the very simple functions of each of the male reproductive system parts. So have a look at that later, but they are there for you. And again, it's really important to at least have a very basic understanding. So if I ever ask you a question about, well, why are these really necessary? What if we just got rid of the scrotum and you have the testes inside the male body? Like, who cares? What a waste of space and time. Just put the testes inside the male body. Actually, that would be very bad for the sperm. And you need to kind of understand, therefore, why the scrotum is there. And obviously, if you remember back, the sperm actually need to become mature sperm at a lower lower temperature than the body is, which is body temperature is 37, and they need a few degrees below that. And if we have sperm in the male body for too long, the sperm actually dies. So that's why sperm in the testes need to be kind of outside the body to be a little bit, the temperature to be a little bit lower. And so that's an example of how you have to justify those parts of the male reproductive system, just to get a really firm understanding of why they are there. Same thing here with the female reproductive system. So we've got the vagina, the cervix, the endometrium, which is the lining. And again, that endometrial lining, that is what, when females menstruate, have a period, that endometrial lining will kind of increase a week before menstruation. And if there is no implantation of a fertilized egg, then that endometrial lining and the egg that is unfertilized, that's in there somewhere, actually is removed, and therefore a period occurs. Then you have two ovaries, and then you've got these things we didn't talk too much about, which are quite interesting, but it's not necessary to know, called fimbriae. And then we've got the connection tube between the uterus and the ovary, called the fallopian tube, super necessary, absolutely important. And then we've got the area that is most important. That's where that is the womb, which is called the uterus. Again, I won't spend any time on this, but they are the parts of the female reproductive system. Please know those for our exam coming up. So then we went through and talked about, well, the two things have to come, the sperm and the egg need to come together, and we call this fertilization. Sorry, that is incorrectly spelt. My apologies. But, we can get over that. That is not the way you spell fertilization. But how does that work? So obviously sperm is deposited as close to the cervix as possible, and then it travels up through a lot of barriers, and chemical and physical barriers that the female reproductive system puts in the way of the sperm. So only the best sperm can survive. Only the best sperm can even get close to the egg, which is great, survival of the fittest. And hopefully about halfway up, at that fallopian tube, then it will meet the egg there and become fertilized, and then as it shows there, the fertilized egg travels down and gets implanted in the endometrial lining, therefore starting to divide, one fertilized cell dividing into two into four into eight, and therefore becoming specialized into different types of cells after we get about 400 or 500 of them, and again into a multicellular organism. That, again, we've talked through this, and if you remember, we did a fabulous movie about the journey of sperm. It was it was a big movie about these kind of people running in little white coats, trying to fight off these nasty white blood cells and other things, these black white blood cells, trying to get them. So if you remember back to that kind of weird, interesting movie, and go through that, that's got a lot of information there that I won't go again through now, but you can refer to later to look at the journey of sperm. And at each point, again, it's a way of minimizing how many sperm get to that egg, and also survival of the fittest, filtering out those sperm that really shouldn't be in the running, because you were once a sperm and an egg that came together, so you had the sperm that won that race. You have been the winner because that sperm was the best one and the fastest to get to that egg to become you. This is a little bit off kilter. So going back the structure of DNA, you can see that going in a little bit deeper, on the right hand side here, you've got your 4 bases, adenine, thymine, that AT, that combine because we have 2 hydrogen bonds that combined A and T, and we've got guanine and cytosine that combine together because it's got 3 hydrogen bonds there. So we have ATGC making the sides, those rungs of the ladder, and we have those hydrogen bonds creating that middle section. All right, so when we want to make a human, when we have that fertilization occurring and DNA is doing its job, well, how do we get one fertilized egg into 2 egg cells and making up you as a multicellular organism, going from 2 to 4 to 8, and 8 into 37 trillion? Well, we need to start making other components of you, and a lot of components are called proteins. So we're going to use the DNA, as we said before, as a blueprint, as a encyclopedia or a code to actually make other things. And so our easiest one to talk about is protein synthesis or polypeptide synthesis, making of, for example, structural proteins to make the sides and edges of each of your cells, which is like the cell membrane. So how do we do that? We firstly have to transcribe the DNA. We just don't take chunks of pieces of your DNA out and use that as the template. We actually copy pieces, unravel that ladder. We use messenger RNA to transcribe the DNA into a new piece of messenger RNA, and then we use that messenger RNA, which is right in the middle there, to translate it into proteins. And there's proteins are made up of, in your body anyway, 20 different types of amino acids, and when you combine those 20 types of amino acids in the right order, then you get any protein you like. And just think, a couple of amino acids, if you rearrange them, even a few, make hundreds of proteins. Think about 20 combinations and permutations if you're a mathsy person. 20 things can be combined in many different ways and makes many billions of combinations of proteins. We don't have that many, but again, using the code of DNA, you can make numerous protein types that make up your body. So that now we get on to kind of a larger section here and talk about traits and characteristics and how do we make you. Well, we're going a little bit beyond now kind of specifically the DNA. When you have a huge amount of DNA, then it becomes a chromosome when you fold it up, and you have certain big parts of that DNA which code, as we said before, for proteins, but those proteins can be elaborated on with a number of parts of that making a number of proteins. We can kind of combine that into a whole section of DNA called a gene. Now, that gene can identify certain really interesting characteristics and traits. Sometimes genes alone don't make up a trait or a characteristic of you. It's many dozens of different parts of your DNA. Many different genes make up a specific characteristic like eye color and hair color, but some are monogenetic, which is just an area in your gene make up one trait, and we'll have a look at these in a second. Okay, so as we said before, you have 46 chromosomes here. It's a diploid number for your dad. Every cell in his body has 46. Mum has 46 chromosomes. When they combine to have you, sperm and egg, well, before that, we have to go through meiosis, which therefore the sperm only has 23 chromosomes. Your mum's egg only has 23 chromosomes in the ovaries, so that when hopefully fertilization occurs, 23 sperm, 23 egg chromosomes make, again, a wonderful diploid number of 46 chromosomes. If you had double that, then we would have a genetic mutations, and then it actually wouldn't start to divide. So all these things now come to the fact of you being you. You're not one big blob of 37 trillion cells. You have many specific proteins that combine to make characteristics of you. Physical traits are the specific ones we'll talk about. So the ones that are inherited, we talked about before, which is eye color and hair color, and whether you've got detached earlobes or not, and whether you can roll your tongue without using your lips or not. And there's a big variety. Charles Darwin, natural selection by evolution, we see variety across our species, and that is due to the combination of the genetics from your mum and your dad, also mutations that are occurring, and different biochemical processes during that fertilization process. So a few things about inheritance patterns. Now we're going a little bit wider and broader. We need to understand a few key words. So when you have when you are inheriting these traits from mum and dad, you can have a trait from your mum or your dad that is expressed, shows up in you very easily, and you go, hey, yeah, I got my nose from my dad's side of the family, or this hair color kind of comes from my mum's side of the family, or hey, my baldness, me, I know I'll probably be bald in the next 10 or 15 years, that might come from my dad's side. Now, I'd say that because a lot of my dad's side, the males of that side of my family are bald and become bald midway through their life. So I would say that that's a dominant trait. Now, when we talk about the genetic and the genes that are associated with that, then the specific gene that does have that trait and characteristic of, let's say a very broad one. This is not perfectly true genetically, but it's a good example. A generic example would be, say, baldness. So if dad had baldness on his side, sperm and egg, dad had baldness on my mum's side, in that egg, let's say her side of the family didn't have baldness, we'd call that allele. So it's the genetic characteristics of that trait that are specifically coded for on that bit of DNA for hair. So whether I lose or not my hair. Now, again, that's not entirely true. Baldness, or having hair or not having hair is, there's many different genes involved with that. But, there's a general idea where all those alleles combined with all the parts of those genetic components combined hint at there's a dominant trait of having hereditary baldness that side. So that's called a dominant allele or a dominant trait. Then we've got those that usually don't show up as much unless you have the sperm and the egg. There are two alleles or two characteristics that have to be less expressed if there is another trait that is greater than that. So that's called a recessive allele. Then we've got a few other things. So we've already talked right down the bottom about genotype, which is the genetic characteristics to alleles. And then we've got the phenotype of how those genes are expressed, called phenotype. Going back up a little bit further, when we have those alleles which are dominant, let's say two organisms, sperm and egg both have the dominant trait of, let's say, baldness, then, that's both inherited from both sides. But if you have baldness and not, then which one is the dominant one will come out. So that's heterozygous, which means they have two different alleles, one dominant and one is not. Let's look at that as a table. So here we can term these and have a capital for the dominant allele and lowercase as the recessive allele. So again, we're talking about a nice easy one is tongue rolling. Can you roll your tongue? Well, if you can without the use of your lips, it's a dominant allele. If you can't roll your tongue, which a number of people can't roll their tongue, I can, but a number of people in our class couldn't. That is a recessive allele. So again, you get one from your mum and one from your dad, and these are the combos that you possibly will get. So again, one from mum, one from dad, capital and lowercase. Again, one from mum, one from dad. So this is a way of saying this is homozygous, which just means they're the same, homozygous, or lowercase t's, two lowercase t's at the bottom here, again, homozygous. But these two capital T's are homozygous dominant, and the one at the bottom, two lowercase t's, is homozygous recessive. And then you've got the one here that's the exact same, capital and lowercase t, or lowercase t and capital T, the exact same one. They both can roll their tongue. That is heterozygous. So can you see the heterozygous ones, no matter which way it's written, because again, genetics doesn't really matter about that, they both have the same phenotypic outcome, which is can roll their tongue. This is how it would look when we talk about fertilization and meiotic division. So that lady there trying to roll her tongue, doing a very good job without using her lips, can still roll her tongue. Now, very funny facial expression, but as we all tried it, it's very difficult to do it without using your lips. If we want to find out mathematically what the possible probabilities of two parents, again, capital and capital, or capital and lowercase t, parent 1, parent 2, what's the chance of parent 1, parent 2's offspring having a child with either being able to roll their tongue or not roll their tongue? This is where we can use this thing here. So we have a way of predicting, which is a Punnett square. So this Punnett square is just a mathematical, nice little visualization. Put parent 1 up the top, parent 2 down the side, and then just combine the two capital T's in this top left-hand box is two capital T's, which is homozygous dominant, and then you fill in the gaps as well. So in this one, you will see that on the top row and the bottom left box, all of those 3 out of the 4, what am I saying? 4 out of the 4 have a dominant allele, a capital T. So therefore, all their offspring, no matter what, 25% there, 25% there, 25% there, 25% there, therefore 100% of the offspring of parent 1 and parent 2 have to be able to roll their tongue because they have this dominant allele of tongue rolling. But there is a little bit of nuance there to say, well, half of them are homozygous dominant, half of them are heterozygous. But essentially, that's genotypic characteristics, when the phenotype is they all can roll their tongue. All right, last few bits. So that's some information you can have a look at later. It gives unpacks what I just said. Now there's some, again, genetics isn't that simple. Again, God's amazing and he's a lot more complex than that. We're just trying to simplify it down to try and understand it with our human brains. But again, there's a lot of variation there. So we're going to just quickly talk about, well, what's the variation to these simple Mendelian traits, which is just a dominant and recessive traits? What are some differences here? So number one would be a thing called not dominant and recessive, but a thing called incomplete dominance, where you have a red flower, and that's where you have homozygous dominant trait, capital R, and then you have a white rose here that is capital W, but when you have a combination, when you cross-pollinate a red and a white flower, just get the pollen from one and put it on the stamen or the other, stigma or the other, then you have the plant that's from that pollination that will be a combination of the two, which is a pink flower. So that's not a dominant or recessive one. That's kind of like both mushed together artistically. So that's why it's called incomplete dominance, because no one dominates. It's just a combo of both. Then you have a thing where, well, they both are dominating in this next one. So here you have a red and you have a white, and they are both expressively, the phenotype expresses both white and red at the exact same time. It's a beautiful flower, and a lot of flowers you see like this are co-dominant, where the nursery, where you buy plants like from Bunnings and things like that, or beforehand, they've fertilized, they've cross-pollinated these plants, and the outcome, the seeds have grown up and they have these beautiful offspring. These plants make flowers that is co-dominant, both having the dominant trait showing up. Then we've got another interesting one where you have a whole load of things going on, and the easiest example here is our blood types, where you have a number of different blood types here, all doing beautiful genetic combos, where you have co-dominance in you. All right, so if you want to talk about co-dominance in us, talking about your blood type, and if you do know your blood type, then that would be a great one to look into. All right, just to quickly, as a reminder, as we talked about before, in humans we have 23 pairs. Just be careful, 23 pairs, but therefore 46 chromosomes. We have 2 sex chromosomes of the 46, or 1 pair. So the sex chromosomes, and then we've got our autosomes, which are all the other ones. So we've got 46 in total, 23 pairs. Then we've got 22 autosomes, and then 1 pair of chromosomes. That being XX female and XY male. That's a little bit more information on that I won't go through right now. Okay, some issues here when we also talking about our interesting non-Mendelian, our Mendelian traits is color blindness. We did do that, so have a kind of little look through examples on Edrollo as well for our example of color blindness. All right, I won't do that one. Here we go. So pedigrees, final bits. So we have another way of showing characteristics and traits that we looked at. One was, again, Punnett squares. The other one was pedigree charts. So pedigree charts are on the next few slides. I'll quickly run through that. Pedigrees show inheritance patterns just like a family tree does, but there's a few specific parts of it. Just like to make a good table and good graph, you have to know what to put in a table and a graph, what makes all those components necessary. And in a pedigree chart, a family tree, a genetic family tree, you have to make sure that you have, for males, we identify males as squares, females as circles. If you have, and this is why we do a pedigree chart, because it's a genetic analysis, a visual genetic analysis of a certain disease or a certain characteristic that you want to follow through your family tree. Now, if it's the worst ones would be, again, the interesting ones would be color blindness. The other one is hemophilia, which is kind of a blood issue. So a lack of blood clotting. So here you could follow your family tree to see who has this blood clotting issue, in your family, and then follow using these pedigree charts, and find out, well, who in of my grandparents who had hemophilia, or this blood disorder, of our great grandparents who had this blood disorder, great grandparents who had this blood disorder. So you might be able to put down who has been affected in the past from a certain characteristic or disease, and then follow that trait through. And you'll be interested to find they'll hint at, well, who did it get passed down through, and was it in your family a dominant or recessive trait? Okay, that's all about pedigrees that we have been through before, so I won't look at that now. Here is an example that you can have a look at later about a dominant trait, as I said before, through a pedigree chart. So you can see generation 1, 2, and 3, and you would be this bottom third generation, and then be able to follow it back through parents and then grandparents. All right, final few bits. We've got mutants. So if there's a mutation, if there is something that everything can affect this beautiful DNA strand, and if it does, then that can cause mutation. It can be just from UV, it can be from smoking, it can just be from normal life and electrons just smashing into this beautiful DNA strand. So those mutations, if it's in sperm and egg, can cause those mutations to be passed on to offspring. And that can also be, mutations don't have to be bad, they can just be changes in that genetic code, and over time, that can cause variation and resilience, and survival of the fittest, natural selection via evolution, kind of Darwinian theory as well. But again, we have to be careful that we're not exposing ourselves to too many mutagens in order for us to have genetic damage. All right, we talked about very quickly some technologies that we could use up at Mars to support life using our new understanding of genetics to make our life easier. Now, the old ones is where it's been used for making bread and making cheese and breeding, sexual selection of the best animal, best female, best male, best cow, best bull, so that the offspring of that cow have the most amount of best meat or most amount of milk. And we looked at some weird and wacky big solid. Do you remember those cows that have, Arnie Schwarzenegger cows, really solid muscles? And then we also looked at these chickens that didn't have any feathers because we don't want them to overheat, and we want to get them to use all that energy into meat production of a bigger bird. So if you remember back, there were some examples of using our understanding of genetic or gene modifications to make more food, or use the livestock that we've got to get more out of them. All right, that goes through what I just talked about here. That's great. So why is it really important? So one, as I said, talked about, more food for us on our plate. Two, we can use it for to have better antivirals, better antibiotics, and also for HIV or cancer patients, or those with cystic fibrosis. We can actually now possibly have gene modification therapies. We also have using bacteria. We have genetically modified bacteria to make insulin for diabetics, rather than putting using insulin from horses, which we used to do, get insulin from them. For the diabetics, we actually have big baths, big vats of bacteria that actually pump out insulin, because we've genetically modified them to do that for diabetic sufferers all over the world. All right, and last one, why do we have to be very careful about us mucking around with our genes and DNA and modifying that? Well, if you might have heard of superbugs. If we modify something, then the consequences of that might turn a little bit deadly, get out into the community, and we can't get that genetic modification back. So we have to be very careful that what we engineer, for example, salmon down in Tasmania, we've genetically engineered salmon to have be nice and big, and so we have big salmon steaks that you can buy from Woolies. But this salmon is in big, massive pens out on the western coast of Tasmania. We have to make sure that they don't get out and then have offspring with the wild population of salmon, because we're combining kind of genetically modified salmon with the salmon that we've genetically modified salmon with the wild salmon that should just be kept wild. So they've got a lot of protocols in place to make sure that they can't fertilize any eggs, or the offspring would die. The other one would be we have to, even though we can have the plants that are able to survive certain insects these days, you can buy certain ones in. Hey, we have to be really, really careful that we're not decreasing genetic diversity. If everyone, for example, there's certain cotton that cotton farmers can produce that is resistant to certain diseases, which is great. But again, only one company or two companies in the world actually have you can buy this from. So all the cotton farmers who want this really good new technology have to only buy it from one company in the world. So that's called a monopoly, and that's not really good if we have one group in the world just holding on to this really, really vital and important biotechnology. So we just have to be very careful about who is allowed to keep this technology and have a lot of legalities in place to make sure that everyone has a fair go with this new kind of manipulation of the DNA. All right, I hope that's helped. Sorry that it took so long, but I hope that's given you an insight into our Generation Martian fertilization and genetics topic, and hopefully you have a little bit of time to use now to just download this PowerPoint that should be on C-Hub, and just use it to look, have a right check, take a few notes, summarize it, or just read through it and try and get it into your head somehow. All right, good luck.